Okay, so this is where we stopped on Friday, right? Energy is the capacity to do work, and so your body needs energy. And the different forms that it uses is heat, light and sound. I don't think your body uses light and sound, but those are forms of energy: electrical energy, mechanical energy, and chemical energy. And we said that the chemical energy that our body uses is stored in what molecule? The mitochondria makes this molecule, ATP, yes. So energy release occurs through the oxidation of glucose. So glucose is the molecule that you need for energy, and it will go through glycolysis, the citric acid cycle, and then the electron transport chain. And ATP is what's formed, and there's 3 phosphates in a chain on the ATP molecule, and the cell uses ATP for many functions of active transport and synthesis of various compounds. So we're going to go through, you remember in biology, I know you take a lot of time studying the glycolysis, citric acid cycle, and electron transport chain. Do you remember that? Yeah, that was 2 years ago. Well, we'll review it. So the first thing that happens is glucose goes through glycolysis, and that occurs in the cytosol. And you produce 2 ATP. And if there's oxygen present, the pyruvic acid, that's also, so you produce 2 ATP, you produce electrons, these high energy electrons, and 2 pyruvic acid molecules. And these molecules would enter through into the mitochondria, and that's where the citric acid cycle occurs. And that produces 2 ATP also, but it also produces these electrons, high energy electrons, that would go into the electron transport chain, and you can produce 32 to 34 ATP from the electron transport chain. But you need oxygen in order to use the electron transport chain. Anything else that... So the energy that your body is stored in the last phosphate bond of the ATP, and energy is stored. What do you think the difference between ADP and ATP is? Right, ADP is adenine diphosphate, so you only have 2 phosphates, and then triphosphate has 3. So you're adding another phosphate group on, and that's where that bond that's formed is where the energy is stored. And so when ATP releases its energy, it's breaking that bond and becoming ADP, and then ADP will get turned back into ATP, and the cycle goes around. So here is an ATP molecule and the phosphate groups that are attached. So ADP is the lower one. So as it goes around the circle, starting at ATP, energy is transferred, phosphate's broken off, and it turns into ADP. And then during cellular respiration, phosphate is attached again, and we get ATP, and it goes around. So glycolysis is the first part of cellular respiration, and it splits the 6-carbon, so 6C means 6-carbon glucose, that occurs through a series of enzyme-catalyzed steps called glycolysis. What did the suffix lysis mean? Remember, it swelled until what? It burst. So burst or the breaking apart. Same with hydrolysis. When we had the hydrolysis reactions was when water is broken up and put into the molecules to break up this larger molecule. The suffix lysis means to break. So glycolysis is breaking apart the glucose. It occurs in the cytosol and does not require oxygen, so it's anaerobic. Energy from ATP is used to start the process, but there is a net gain of energy as a result, and there is the net gain of 2 ATP. Once the pyruvic acid that's producing glycolysis enters mitochondria, then it begins the aerobic respiration. It occurs in that mitochondria, and there's a lot more ATP that can be produced with aerobic respiration than anaerobic. So the final products of glucose oxidation are carbon dioxide, water, and energy. This is showing how the simple sugars goes through glycolysis. The proteins and fat skip that glycolysis stage, and they're broken down into pyruvic acid, and we'll go into the citric acid cycle here, form ATP, electron transport chain, forms ATP also. Wow, you can't see that very well at all, can you? Can you see it better on your chart? Well, it's in your book, and it's a figure from your book. So the enzymes controlling the anabolic or catabolic sequence of the reactions must be in a specific order, and that sequence is called the metabolic pathway. And a lot of times, those enzymes are in that order in the cell membrane or wherever they're located. And the rate of the metabolic pathway is determined by a regulatory enzyme responsible for one of its steps. And so it is called the rate-limiting enzyme because it determines the rate that the reaction can occur. And it's a lot of times the first step in the reaction chain. Okay, so this is over the cellular respiration. Hold on, I didn't plug this in.
OA transferred to a carbon molecule. COA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Which is what we called the citric acid cycle in that one diagram. Yeah, that one. So right there. And I don't know that you need to know all those details with the NADH and the FADH too, but it does produce 2 ATP. It does have to go around twice in order to, because you produce 2 pyruvates or 2 pyruvic acids, and then it has to go twice around with each of the, well, once around with each, so twice total. And then I think this one's just on the glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, so that's one part that wasn't in the notes, that if there's no oxygen present, the pyruvate, instead of going through the citric acid cycle or the Krebs cycle, and getting transferred into more ATP, it's going to get converted into lactic acid. And these NADH molecules, those are the high energy electrons that was on the one chart that had high energy electrons going out and down to the electron transport chain. That's what is going to go into the electron transport chain. Okay. Okay. And I think that's all we were going to do today. So tomorrow we're going to look at DNA and then protein synthesis.